this was, I gave this talk at the horse fair last year, so it was specifically towards breeding then, but really this goes into the genetic testing is important, I think, you know, in making decisions in buying a horse, particularly certain breeds that are predisposed to issues genetically that we know that tests are available, quarter horse in particular, Arabians. It's becoming more and more widespread. So it's not only the breeding, buying horse, but actually if you have a horse that might be a carrier for one of these diseases too, it also helps us identify those and helps us make decisions clinically and how to manage them best. And that's on an individual basis or a bird-wide basis. So, you know, in 2006, and what allows us to really get sophisticated with this genetic testing is the horse genome was mapped fully at 97 or 98% in 2006. And the genome is just a complete set of DNA of any individual organism, but that's the horse. And this was done as an international effort. A lot of it was done at the Gluck Center in Kentucky, but all over the country. And they identified about over 20,000 genes, and that's increasing now as they get more sophisticated in mapping these things out with all the computer system programs and such. The neat thing about it is that all that information is available to researchers all over the country, so and they can pick out their specific research interest and go with that. So we're really getting an explosion of information right now in the genetic basis of many of these diseases. Right now, commercially available, there are 10 syndromes that we can test for. And we'll go into the types of diseases that we're talking about. And they're identifying these mutations. What you'll find, and we'll talk about a little bit, is that's kind of the black and white of it. These are the easy single gene diseases that they're picking up. But they're getting a lot of information in terms of what's a predisposition mean and what are the factors in the environment that turn on these genes to cause these problems, and we'll talk about that. So it's pretty neat stuff. And they're following the lead of a lot of what they've done in human medicine. And we're behind, but we're doing the same type of thing. So as I alluded to at the beginning, certainly in determining what sire is going to be bred to what mare, we can make some intelligent decisions based on you know, what our risk level is that we're comfortable with in terms of having offspring that may or may not have a genetic issue or be a carrier. Again, as I alluded to, and you know, I'll give you an example, HYPP, I think most people are familiar with that in quarter horses, where they can have some very serious and often fatal complications. If we know that horse is HYPP positive or positive, then we're going to make some different decisions than we would if we were working in the dark. Okay, so that's important in that. And then in some of those conditions that are less black and white, the horse might be a carrier or might be mildly affected. It allows us to kind of alter their environment relative to nutrition, levels of stress we're going to put them under, that type of thing, so we can manage them appropriately. So obviously, I think it goes unstated, the goal of all the testing would be to eliminate genetic disease. It's out there right now, and one of the things is that since the early 80s, when we've been doing a lot of ship semen, artificial insemination, embryo transfers, it's been great because it's allowed a lot of people to tap into a genetic pool, but it's also narrowed the genetic pool. We've had several, you know, you can probably name a dozen quarter horse stallions, for example, that have bred literally thousands of mares. And so while it helps propagate those good characteristics, it also is the bad characteristics that go with it. And that's why we see, and part of the reason is the quarter horses and the Arabians, both two more popular breeds, we're seeing a lot of these diseases that have been identified and have been concentrated. So, and we'll talk about those, not in detail, but we'll go through a mention on them. Again, the bottom line, it really is to know what your risks are, assess the risk, and find that level of risk that you're comfortable with in terms of managing your herd or buying an individual animal or breeding an individual animal. And then again, not to be redundant, but when we know this information about a particular animal or a group of animals, we can certainly tailor their preventative health program around that to minimize the chances that we're going to have a clinical problem. So now this is the time to take a quick nap. Everybody remembers high school biology, but I think we have to talk about the basics just really quickly so everyone's on the same page. In a horse, they have 31 pairs of what we term autosomal chromosomes, not the sex chromosomes. And each one of those chromosomes has thousands of genes on it. And each one of those genes has two forms, or alleles, as we would call them. One comes from mom, one comes from dad. Very basic. So when in any of those genes, if there's a mutation, the DNA sequence is changed or altered in any fashion, what they would term a point mutation, then that can result in an abnormal biologic process. You may or may not get a disease, that type of thing. So that's the basis of just the real basics. Just schematically, you know, you have the dam in the blue, 2 chromosomes, there's a gene on either one, so that's the alternative, or the forms of the gene in the little black, and so one, okay, straight transmission, one from the sire, one from the dam. So it's important to talk about the types of inheritance that we have with these guys, and many of the diseases we deal with are autosomal dominant diseases. In other words, they only need 1 copy of the bad gene to cause that disease or produce that defect. When we have autosomal recessive disease, it takes 2 copies of that allele or that gene, the same, to produce the effect. So that would be the carrier state if they just have 1 of those. Some cases they are clinically affected, but not to the degree that one that held the 2 traits. Okay, and that depends on the condition, and there's a lot of other factors going into it. So it's the basic Mendelian genetics. It's the old. They were looking at the colors of the peas, you know, and the peas were different colors, and they figured out all these crosses. So straight inheritance. So again, trying to break it down even a little bit further, most of the tests that we do today that are available, and there are more online, this is a very fluid situation, there are more and more coming all the time, are what we would term single gene diseases. There's 1 gene that controls the production of that defect. And we'll go over those 10. I think more interestingly, and is where this thing is going as we get more sophisticated, is that there are, and the majority of these genetically based diseases are polygenic. There's multiple genes involved, and most of those genes and their expression of those genes is modulated or altered by the environment. And again, nutrition is number 1, level of stress, level of exercise. All these things come into play, whether that bad gene, if you will, talking about a disease, is going to be manifested or not. Obviously, because of the complexity, they're more difficult to have a black and white test for, but they're getting more sophisticated in terms of identifying these genes and identifying markers of these genes that will allow us clinically to have some tests that we can run to see if these animals has the chance to be effective. So, very simple protocol for testing. UC Davis is probably the most popular site or lab to send things to. And you can get online and look through. It's actually very informative. They have a nice website to look at. They offer, I think, 10 or 11 of those tests. University of Kentucky, the Gluck Center, again, they were very active in the Genome Project, and they're doing a lot of the testing. University of Minnesota is doing a lot of genetic testing for the muscle-associated diseases, PSM, those types of things, chronic tying-up syndromes, those things. So we can use anything that has DNA content, anything that has cellular content for the testing. You know, commonly in the registration kit, you'll get a little envelope that you're going to pull mane hair, tail hairs in, and you're going to look at the roots of the hair. That's a basis of cells. You can use blood, semen, etc. We've, even had frozen semen on stallions that have been long deceased that we've sent in and had some of these tests run. So it's anything is fair game. It's kind of like that CSI, where they do the little swab and all that other stuff. Interestingly, and we did a little of this when I was down at Minitube here, the embryo biopsies, where we actually look at a small sample of cells from an embryo early on, day 6 or 7, and then freeze that embryo, then we can send it in for testing. And if that comes back clear or it's not a carrier or anything like that, then we can go ahead and use that to hopefully get a pregnancy. They're doing this in people quite commonly. It's called pre-implantation genetic screening. So it's very commonly done, or like people, women that are over 38 or 39 years old, they're worried about genetic issues, they'll go ahead and do this type of thing. And they're actually not taking cells from what would eventually turn into the baby or the foal, but it's the cells that would eventually become the placenta. So it doesn't damage the embryo itself that way. So bottom line, is readily available. There's a lot of actually private companies that are getting involved in this too. Most of them, in one way or another, associated with the universities, and the cost is not prohibitive at all. You'd be surprised as this goes on how cost effective it is. So when we send the test in, then we get this sheet that comes back, and it's going to tell us, what the risk is of this individual animal being a carrier or being affected. And it's important to distinguish between what we term homozygotes, that is, they have 2 alleles that are the same. So, for example, if they're both normal, so NN would be reported. If one is a carrier, for example, only has one copy of that defective gene, then it would be a heterozygote, and one normal, one defective, and as you can see, DD both defective. This classification allows us then, just by basic probability, to determine the risk that an individual animal is going to be affected or be a carrier or be non-affected. And increasingly, and Quarter Horse Association is at the forefront of this. In the next several years, you're going to see this going to be a requirement on all animals that are in the breeding population to have these tests run, and they'll be listed right on the registration certificate. Right now, they do that for HYPP in quarter horses, and in fact, by 2017, the quarter horses are going to require all mares and stallions to have a series of 5 tests done. The Arabians have been discussing it for CA and SIDS, so cerebellar abiatrophy, and SIDS are the most common genetic issues with those guys. Some of the draft registries are too. So anyway, it's going to be a requirement. The quarter horse, I think they run, there's 5 tests they run, and I think it costs $80. So it's not too bad at all.
So will they eventually get to the point where if they're testing positive for things, they won't accept foals to be registered?
No, they're not going to get to that. I don't think they'll get to that point. It's going to be, because I think that would be too draconian. They're going to essentially, I mean, if you've got a carrier for something that's bad, or it's affected, that's not a not lethal condition, then no one's going to breed to those animals, or they're going to be worth less on the market. That kind of. So I don't think they'll get to the point where they'll be non-registrable. Yeah. So 2 types of diseases then. We talked about the autosomal dominant. That is, you only need one of those bad alleles to form the disease. When we test this out and it comes back, if you're going to breed an animal that carries one of those bad alleles to a normal animal, 50% of those animals, and this is basic probability, are going to be affected. If you have 2 animals bred together and they both carry that bad one, then 75% of those animals are going to be affected. And then, of course, if they both have 2 copies of the bad gene, they're all going to be affected. And we'll talk about the types of autosomal diseases we test for right now. And here they are right now. HYPP, hyperkalemic periodic paralysis, quarter horses primarily, paints 2. A muscle condition, polysaccharide storage myopathy, seen not infrequently in quarter horses. The warm bloods are affected, drafts, other breeds too. Malignant hypothermia, again, which is a muscle issue that we see in the paints in the quarter horses. Those 3 are going to be on the AQHA list of 5 that are studied. I think interestingly, the gray horse melanoma, the gene, the dominant gene, big G, when you have a homozygote, they have 2 copies of that. You know, all these horses, it's very rare to see a teenage gray horse that doesn't have a melanoma of some sort somewhere on them. And we know that when they have 2 copies of this dominant gene, their graying rate increases. So all of a sudden, you know, it's these, what you normally would see a 25-year-old where they look white almost, right? They're going to be white earlier, and they're going to have a higher incidence of melanoma and more invasive melanomas. Then, of course, it's only carrying one of those. So it's something you can look at, and particularly if you're breeding and you have some gray stock and you're breeding that, you might want to.
Do they develop the tumors at a younger age?
Yeah, they do. Yeah. Very interesting, and that's an example of a color-associated genetic disease, which we'll talk about here in a little bit. But you know, people are breeding for color. They're breeding for color pattern, Appaloosas, paints, even some of the Rocky Mountains and things like that. And then sometimes breeding for color has unintended consequences in terms of concentrating some of these diseases. Recurrent exertional rhabdomyolysis, and this is a tying up type syndrome in thoroughbreds particularly, but seen in standardbreds, Arabians. This is something that's not a straight autosomal dominant in the sense that the environment, particularly stress, really has a factor in how this is manifested in the horse. So those are examples, and they can all be tested for. On the flip side, we have autosomal recessive diseases, and that is where you have to have 2 of the copies of the bad gene to get the defect, right? So if we have a carrier, and that would be the one that just with 1 copy of it, a carrier bred to a normal animal, not a carrier, half of those are still going to be carriers, the other are going to be non-affected. And you can see go down the line. what your probabilities are. And you can use this to kind of figure out where you're at and what, if you want to take that risk of breeding these animals. You know, if you're buying a brood mare, you're looking at a stallion to breed to your mare, and you know your mare's genetic status, you can use that information. Is it really worth the risk to go to this particular stallion? Examples of that, Herda in the quarter horses. This is another one of the quarter horses is on their list of 5. This is a skin condition that we commonly don't see until they're 2 or 3 years old and they go into training. But we're not going to get into the specifics of the disease at this point. I think it's out of the realm here. In foals, it's something we don't see very often, but lavender foal disease in Arabians. Cerebellar pleiotropy, which is a neurologic condition in young Arabians. Getting back to what to me is very interesting from a clinical basis, it's the polygenic, the diseases that are, it's an interplay of multiple genes that are causing these problems. It's the additive effect, if you will. There's a lot of modulation or influence of the environment on the expression of these genes and hence the clinical presentation of the horse. They're difficult to characterize, and because of that, they're more difficult to get a test for versus the single gene diseases, but they're equally important, I think, in terms of the economics and making decisions on how to manage your animals. And again, this genetically mediated susceptibility, you'll hear us say that this animal is genetically prone to a club foot, for example, or something like that. Okay, so this is kind of what we have those genes there. What turns those genes on? That's what we want to identify, or what genes are markers that we can identify to give us a heads up and a head start on managing these conditions. Some examples of what I'm talking about. Recurrent airway obstruction, kind of a Hebe type condition. Certainly these animals, these are the ones that you talk about, the environment, you know, these animals are fine in the pasture. They're genetically prone to an issue. You get them into a dusty barn or indoor conditions where the airflow is not good and they're going to be symptomatic for you. Very popular topic, everybody's talking about equine metabolic syndrome. We certainly have identified this. We've identified what we call a phenotype, or the outward expression of what these animals look like that are prone to this. But they're starting to work out the genetic basis of this and how maybe we might be able to turn some of those genes off so we don't see the bad sequelae that we do sometimes founder and things like that. Interestingly, susceptibility to certain types of infectious disease is a polygenic thing, and they've identified a gene in thoroughbreds and Arabians that's not a test available for it yet, but we always talk about these animals that are thin-skinned and they're really getting eaten up by the mosquitoes or the gnats and things like that. Well, you know there's a genetic basis to this. Not that their skin is thin, but there's a genetic basis for this and why these animals react like they do. And you know, looking ahead to the future, the whole goal is to see, we're identifying these animals, we're testing them, then how can we change it? How can we turn off those genes? And that's where medicine is going, which is really quite deep. The most common thing we probably see are orthopedic problems. Obviously, conformation is passed on. We know that. The orthopedic disease complex, again, OCD in the joints, osteochondrosis dissecans, clubfootedness, or the flexural deformities that we see not uncommonly, lordosis, the sway backs, you'll see in the saddlebred some. In the thoroughbreds, some animals in certain lines are really prone to superficial digital flexor tendinitis. They pull their tendons very easily compared to animals not carrying those genes or those markers. Suspensory ligament degeneration in a lot of the gated breeds, the Pasophinos, the Passos, that type of thing. And really simply the development, OA stands for arthritis, osteoarthritis. And just like in people, there are certain animals and certain lines that are more prone to develop arthritis at an early age without any obvious inciting factor. And we think that's genetic. And what turns that on? That's the underlying question. Interesting for me, and we do know, we've identified over the years, there's certain lines that are more fertile than others. Mare side, mares and daughters, it goes along. There's a genetic base for that, obviously. What is it? How can we identify it? In terms of making smart decisions in purchasing animals or breeding certain animals, and how much we're going to spend on that. Certainly in certain lines of stallions, we're not going to pick on the Arabians, but we know that a lot of these Arabians, by the time they're 10 or 12, have a very fast declining rate of fertility. And again, there's been a lot of inbreeding and line breeding in the Arabian breed, and we're passing that on. So a lot of these very popular stallions are subfertile, and it drives you crazy because you're trying to get babies in there, you know, and then turn your hair white. That's a color associated genetic.
But it's not your fault.
It's not my fault. It's the environment. Examples of color-associated genetic diseases. I think most of you are familiar with the ovariolethal white syndrome, which is the white hair coat, and it's associated with a non-developed intestinal tract, if you will, in these babies early on in their life, colic badly, and it's a fatal disease. It's a lethal disease. So we want to be able to identify those carriers and avoid crossing the animals that are at risk, just to avoid the problem, obviously. Very interesting syndrome is the ocular anomaly syndrome in the Rocky Mountains and Kentucky mountain horses, and it's just kind of a whole host of ocular problems, iridial cysts and things like that these guys have. And this is strongly associated with the silver coat color, which is very popular. That's what they're breeding for, that's what they like. And the unintended consequence of that is this problem we've propagated with the eyes. This is one of those that, you know, the homozygotes that have got the 2 copies of the gene, they're affected more severely. Interestingly, if they just have 1 copy, they're affected, but to a lesser degree. So, you know, again, it's a management issue, and this certainly comes into play when we do purchasing samples in these types of animals. It's very rare to see them that without an issue of one or another. As I kind of alluded to earlier as well, many genes have effects on processes in the body that are totally unrelated. And this is how the color, it's called a pleiotropy gene. And when we get that one gene and it mutates, and maybe it mutates to give us the color that we want, right? And or the pattern that we want. But it might also affect, for example, in ovariolethal whites, development of the intestinal tract. So, you know, it's one of those things, there's nothing, I'm not against breeding for color or pattern or anything like that, but we have to be smart about what we're doing. And the more information we get, obviously, the better off we're going to be. The two-edged sword, assisted reproduction, by that I mean artificial insemination, embryo transfer, the in vitro fertilization, the cloning, all that stuff. We can use it as a tool to maximize the genetic diversity because we can make smart decisions and things like that, and we can propagate it that way in terms of numbers of animals bred, number of stallions used, that type of thing. But really what's happened over the years is that we really amplified and blown up the incidence of a lot of these diseases because we've concentrated all those bad genes just like we've concentrated the good genes, the performance attributes of some of these animals, their conformation, all that stuff. There's a flip side, so there's both sides of the coin. Again, you know, a lot of it can be traced to this excessive breeding of a few popular stallions. You know, the impressive disease HYPP is the #1 prime example of that. It's not something we see as often anymore. We do see some of it, but because people are making smarter decisions, and they've been testing for that for probably 8 or 9 years, I would imagine, something like that. So, factors to consider in testing. Obviously, you know, the #1 thing is when we're buying animals or breeding animals, we want to consider their genetic background and makeup. I mean, I did that, and I don't want to be redundant on that. The other thing to consider, I think, is the quality of life of these animals. Some of these animals, if we know we're breeding for them, we're willing to take a risk, we know. But some of them, if they're just carriers, for example, or they're just mildly affected, you know, what's their quality of life? Could we avoid putting these animals through this type of thing if we don't have to, if we can go a different way in our breeding? And a lot of that depends on how lethal that condition is. Is it going to turn into a chronic condition that's hard to manage, that's expensive to manage, and that's not fair to the animal? I mean, we need to consider that type of thing. The biggest thing on these, in this genetic testing, in terms of eliminating genetic disease, is the role of the carriers. These are the animals that just have 1 copy and they're not clinically affected. If we can get rid of all those, then obviously we're going to get rid of the disease, because the affected ones, they kind of have the scarlet letter on them anyway. You know, so the goal of all this is to identify carriers, and that's, unless they're going to be a gelding, you know, then we need to get them out of the genetic pool. So, you know, some of the roadblocks, obvious practical roadblocks, is that there are some owners that have valuable animals that might be carriers or stallions, and they don't want their animal tested because it's obviously going to hurt their market value. So they're trying to get over that hurdle, and that's why AQHA, for example, has kind of, in stages, put this testing into effect. And And certainly, you know, there's certainly some ethical and some legal considerations. And if you're testing and you know it, you have to disclose it. You know, so it's better just to close your eyes and say, I don't know. Also very important is how do we manage the unaffected carriers? And, you know, again, our whole goal is to identify these carriers, get them out of the breeding pool, and then we can slowly start to whittle down the incidence of these disease studies. A lot of people still do breed their carriers, and they know it, and hopefully they're making the choice to breed to a non-affected or non-carrier. That doesn't always happen, and a lot of people roll with us. That pretty much says everything that we've kind of talked about. That's all I have.
Do you know if some of the, well, HYPP is a great example of a disease that was rewarded in the show ring, essentially, because the stallion that was responsible for it produced halter winners.
Right.
Has the Quarter Horse Association or the Arabian Horse Association started changing their judging criteria to try and weed these guys out?
No,
of the show
no, they haven't. And I think that's it's going to be a long process,
Yeah.
you know. But until, you know, fortunately, this is, you know, we've been able to do a lot of this since the mid-2000s, so 2006, 2007 on. So we're starting to reach the end of the breeding life, useful life of some of these animals that are affected. And hopefully these breeders are being smarter in cycling in the animals that aren't the carriers and that type of thing. But it still goes, you know, I mean, if an animal is performing well, it's a Grand Prix animal, whatever it is, and they're still going to breed it probably. But I think the value lies, at least at this stage, is just identifying and knowing what you got.
How long has this herd been around, per se?
I would say, just off the top of my head, I would say it was first identified in the early 2000s, probably. And that's, you know, the cutters and reiners really exploded about that time, maybe a little bit earlier, late 1990s. And the value of these animals went up, and they were just breeding. I mean, you would not believe the number of animals that were bred to certain stallions. And all of a sudden, we concentrated all those things in. And there's a lot of line breeding that goes on. You know, father, daughter, you know, this whole thing. So, you know, interesting. There's some other stuff, and they're not commercially available now, but I think it's a French company or a Dutch company. They've identified a gene or a marker gene that, with great degree of accuracy, can predict which animals are going to easily gate versus non-gated animals. And yeah, so it goes down to that level, you know, once they're starting to really pick these chromosomes and these genes apart and looking for those things. So, you know, obviously, if you're looking for a gated animal, you know, you want to optimize your chances of getting one. So that's just an example. There's a bunch of those types of things that are going on right now, and they'll be available. Not necessarily at the university level, but they'll certainly be available. You know,
is that quite an expensive test, though?
You know, I don't think that is. You might even be able to get a link on the UC Davis website to that type of thing. I think it's a couple $100, something like that. As I said, the price of these is going down because once they get the basics and they're set up to run these tests, it's really very cost effective. I mean, you're talking 25, 30, 40 bucks a test.
Exactly.
So eventually, will they be able, let's say you have a horse and you can't figure out what's wrong with it. Will they be able to test her genes of a horse to find out what's wrong with that horse?
Well, not necessarily, maybe not in that context, but I think where this is going, though, is that the more information we have on what genes are involved and that type of thing, you've all heard, it's been in the papers in the last several years, of the idea of gene therapy, where we're going to turn on certain genes that are beneficial and turn off other genes that are detrimental to the individual. So whether it's heart disease, whatever, that's how, even treatment of arthritis, rheumatoid arthritis, is turning off certain genes. So in that sense, if we can identify the problem, we still have to come up with the problem, we still have to have a diagnosis. And I don't know that we're going to be at the point where we can just do a simple genetic test and have us tell that, you know, her hock's sore or something. Yeah.
Where are we at compared to like overseas breeders? So Germany, or are we kind of at the same level, or?
I think we're a little bit more sophisticated.
Are we? Okay.
Yeah, we are. Yeah.
Standards a little bit getting higher. That's
yeah, it is. Yeah.
Pat, what
Yes.
diseases are out there that are commercially available for the test?
Verda, HYPP, malignant hypothermia, glycogen branching enzyme deficiency, which is a muscle disorder, PSM. Those are primary. And you'll notice that most of it is heavily weighted towards quarter horses, right? I mean, that's the biggest breed that we're dealing with. A condition, a skin condition called junctional epidermolysis bullosa, or JEB, and Belgians in particular, but it can be seen in saddlebreds and certain other draft breeds. That's available. The Overo-White syndrome, lavender foal, in Arabs, cerebellar avotrophy, SCIDS, which is a combined immunodeficiency in foals in the Arabs. I think there's 10 or 11 right now that are commercially available on a widespread basis. There are certain labs, because they have a research interest in it, if you get in contact with them, they will explore and run some tests for you, but these are not, these are kind of on an as-is, hit or miss basis, you know, type of thing. So it's not a commercially available thing, but the more information they get that way, they can try to relate their research findings to clinical findings. Yeah.
Is the Quarter Horse Association or anybody looking at like juvenile arthritis in quarter horses?
You know what, I don't know. That's a really good question. I would hope so. And the first thing they would do is just burn all the lunge lines.
There's an alphabet for
Yeah,
trees.
right. But again, that's an example of, you know, and that's a breed we see a lot of that in. That's an example of the environment modulating the expression of the condition. If most of those animals weren't, and I'm not, because it's not black or white by any means, but if a lot of those animals weren't on a lunge line for 3 or 4 hours a day, that type of thing, several times a week, and we're adding that stress to them in the sense of physical stress on the joints, then we wouldn't be seeing the expression of that a normal young horse wouldn't be showing. So it's very interesting, and it all really comes back to it is a genetic predisposition for certain things that we're seeing, but we're just being able to identify it now rather than shooting in the dark.
So are there labs that we can send samples to that are doing experimental stuff that we might be able to get some information out of?
For example, Wahlberg's lab in Minnesota. You know, they're running PSM and GBED and all that stuff. I was told, and I haven't sent anything up there, but they have several other research projects going on that they're. And I don't know that I don't have any specific syndromes, but that would be a good example of one. You know, Davis is doing a ton of it. Gluck Center is really, really active in that way, and they're getting a lot of money from the thoroughbred industry and such like that. Yeah.
I was surprised to hear you say that for those color-associated diseases, that the genes that are underlying the color are also responsible for the diseases. Is that actually really known, or is it more likely to be linkage to a gene that's causing?
It's probably linkage to a gene, but there are some genes that control multiple processes.
Sure.
Right.
Curious, out of the Welsh breed, are they known for any specific kind of common diseases in their breed?
No.
I haven't really heard very much.
I know, and I don't. Nothing rings a bell with me, Lisa.
And they might be prone to some of the heavier, bigger Welsh cobs, maybe prone to some of the muscle disorders that the draft types are prone to. But other than that, I haven't heard of them.
No, I haven't either.
She was asking about laminitis, but I don't think.
I've not seen.
Unless it falls into that polygenic
Right.
predisposition towards
Right.
metabolic syndrome.
Right.
For people
people, including as part of pre-purchase exams for
Some of them are, yeah.
stallions and brood
Oh,
mares?
yeah, absolutely, yeah.
Well, sometimes we get asked to run them for, not necessarily for breeding, but for, just to know if, you know, if they're gonna have a muscle disorder down the road.
Right.
You know, do we know that now before they're going into work? So do you wanna buy that horse? You know,
Or do you
maybe a
wanna buy-
gelding, but they still wanna know.
Yeah, or do you want to buy the horse, or if you're going to buy that horse, now
How
we
do
know
I feed it?
how we're going to manage it,
Work
how
it.
we're going to
Yeah.
alter its diet, and we're going to do various things in terms of getting them in shape, in terms of their workload, that type of thing.
Yeah. When HYPP first came out, a lot of people wanted to know if they had heterozygous carriers, because some of them were symptomatic.
Yeah, and
wanted
that's
to
definitely,
know
and
that-
that's one of those diseases, that's an autosomal dominant disease. But the homozygous with the two bad alleles are severely affected, but there's various gradations. There's a wide range of clinical signs that those heterozygotes show. Yeah. Yeah.
So clinically, as far as how many clients do you get that would ask about genetic testing, like as part of their breeding programs, and is it the responsibility of the owner or responsibility, well, it's responsibility of the owner, but do you guys send out the samples or do they send them out themselves?
Generally, we'll send them out for them. The Arabian breeders seem to be more interested in this than the other breeds. I mean, we do a lot of warm blood stuff too, and there's not any, you know, it's not a really wide, you know, a wide issue because there's such a genetic diversity there. And of course, the quarter horses, they need this testing to be registered. Yeah. But the Arabians, I think, are the number one. They're really worried about cerebellar pleiotropy and skids. Those are the two ones they're really concerned about. Not so much the louder the foal. But anyway, that was, I mean, just kind of a quick summary of that. I just think it's good to have it on your radar in terms of, you know, going forth. If you're going to be in the industry and you're going to be buying horses or breeding horses, that type of thing. Just add a bit of information for you to make a good decision. Cool.
Thank you all for coming.
Yeah, thanks for coming.